Hey guys, welcome to Cosmic Solutions, Dr. Priyanka's Biology, where we discuss everything from biology to biotechnology. So today we shall see what is bacterial transformation. So if you can recall, I had made a video for the 12th standard students where we had discussed about genetic engineering and we had discussed in short what is transformation because just the term was introduced over there. So I thought of making a special or separate video of bacterial transformation, which will be very useful for those of you who are interested in biotechnology or who are pursuing biotechnology, because the most of our things are based on the principle of bacterial transformation. Okay, so let us try to understand the term transformation. Transform, the English meaning. Okay, the literal meaning of it is to change, to make certain changes. Now, when we are talking about bacterial transformation, okay, the thing is we are making changes into the bacteria. Now, why are these changes made? These changes are made so that some useful product or the product in which we are interested should get produced or should get expressed by that bacteria. So this is why this transformation is being done. So now let us come to the definition of this bacterial transformation. The process in which we are making a bacterial cell to take up a foreign DNA is called as transformation. Okay, we are making some deliberate changes so that the bacteria is able or is forced to take up the foreign DNA, and in turn this foreign DNA that we have, okay, it will be or it will have a particular sequence which will be useful or which is responsible for producing some sort of product in which we are interested. So this is what is bacterial transformation. Alright. So now let us see the process. Now as we had discussed before, okay, bacterial transformation. Now here we have two components here. One is the plasmid and the second one is the bacterial cell which is carrying the chromosomal DNA. Now this plasmid, okay, it is the foreign DNA. Now suppose we make changes into the surface of this bacteria and we make this plasmid cell to enter inside this bacterial cell. The process is called as transformation and the product what we have is called as a transformed cell. Okay. Now here we have taken example of just a plasmid. But what if we introduce, because we all know that plasmid is a vehicle which will carry a passenger, and here the passenger is in form of some gene. Now suppose this red portion what I have done here is it is producing say some protein called as protein A, and which is having some commercial use. Okay. So when we introduce such a plasmid into a bacterial cell, what we will what we will look for is the expression of this particular foreign gene, which in turn, okay, when we multiply such a cell under culture condition, so many times, okay, that many fold protein of this particular newly introduced gene we will get as the product. So this is what is your transformation. Okay, now let's get moved on to the next slide. Now here we have some piece of DNA. Suppose now this small section of it is responsible for producing some gene. Okay, it is responsible for expressing some protein or some useful product. And here we have the plasmids. Now our intention will be to cut only the specific required portion of that particular gene which will have the gene of interest, and then introduce into the plasmid. So before that we need to make sure that there are cuts produced in the plasmid as well as in the gene portion carrying the gene of interest. We need to use the same scissor, that is your molecular scissor, that is restriction enzyme. So based on that, specific cuts will be produced in both the cases, and after this, okay, they will be brought together, okay, and they will be sealed or they will be glued with the help of ligase. There will be ligation, and such ligated, such ligated plasmids, now they are called as recombinants because they have undergone recombination, right? They will be transferred into, or they will be forced into a bacterium, okay, and that process is called as transformation. And now this cell what we have, it has undergone transformation, because if you see there is this plasmid yellow thing has entered inside this, so we call such a thing as a transformed cell. Now this transformed cell, if it is carrying the newly made up construct which is having the plasmid DNA as well as the gene of interest, we will have a recombinant transformed cell. And the other thing that is possible is, now for example, this plasmid has resealed again, that is, it has undergone self-ligation so that it is not having the gene of interest. Okay, that is condition 1. So this will be your non-recombinance. Second case will be after transformation, wherein there will be some cells which will carry the recombinant gene. Second condition, when there will be, they will carry the non-recombinant gene, because that is also transformation, right? And the third thing possible is that there is nothing that has entered inside, no plasmid has entered inside. Now this becomes a case of non-transformed cells. Okay, these two conditions will be your transformed cells, and this condition will be of your non-transformed cells. Okay, so now let us go to the next part. Let us try to understand this now. Now, first of all, to undergo transformation, your bacterial cell should be able to take up that DNA. Okay, and this ability of a bacterial cell to take up foreign DNA is called as the competence. Okay, the ability of a bacterial cell to take up the foreign DNA is called as the competence of the cell. Now, if you see naturally, okay, naturally the competence of the cells is very low. So that means further the efficiency of transformation will be reduced, right? So because of this reason, what is done is we try to make a cell competent artificially, or using some method we try to make certain changes so that the bacteria becomes a competent cells, and it will allow that foreign DNA to enter inside the cell. So how does this happen? Okay, so this can be done in different ways. Either we can carry out certain changes by using, some electric pulses, or giving some sort of shock, or using some chemicals. Now why this is required? Okay, why this happens? Now if you see the exterior surface of your plasma membrane, it is negatively charged. Now this happens because of the phospholipid head groups, okay, which imparts a negative charge. And if you see, your DNA is also negatively charged. So overall what happens is there is repulsion. Okay, your cell is not allowing your DNA piece to come towards it or come near it, because it is both the things are having negative charge. So there is repulsion. So that is the basic thing. So we have to make some changes in this cell so that it is easy for the foreign DNA to enter inside the cell. So what is done is we do something called as a heat shock method, or a chemical method in which we are using the calcium chloride. Okay, we use divalent cations of calcium, so in form of calcium chloride. So normally this calcium chloride has to be ice cold, because it is seen, many of the studies they have seen that ice cold calcium chloride increases or enhances the chances of competence. In this picture, we have E. coli in the log phase. Now, reason for taking cells in the log phase is that, okay, that is the best metabolic phase. Okay, normally it is mid log phase, because in this phase the cells are very active, they are metabolically active, and the efficiency of transformation what we will get will be very high as compared to your any other phases. So here what is done is, normally we see that the OD at 600 nanometers, it should be somewhere around zero point 0.4 to zero point 0.9. So that is the best ideal situation where we can take out or when we can harvest the cells. Such cells are taken and they are centrifuged. This liquid is centrifuged. Now after centrifugation we will get the pellets. We will get the pellets. We will get the pellets which will have the cells, these E. coli cells. So after this they will be resuspended in ice cold calcium chloride. Now here what happens is, this calcium chloride, okay, it enhances the permeability. It enhances the surface. So which is going to help the process further. So after this, again these cells are resuspended, or they are they are made into aliquots. Okay, they are redistributed into smaller append of tubes, and here plasmids are introduced in this append of tubes. So here if you see, there is a tiny yellow circle indicating a plasmid. Okay, so after introduction, what is done is, we give a heat shock, okay, of say 42 degrees Celsius for a very short duration of say 30 seconds. Again, this time duration it will differ. Again, it will depend on the volume, and again based on the literature survey we have to decide. Okay, so it will be mostly it is 42 degrees Celsius for a period of 30 seconds. Okay, this is the ideal condition that is used. So after this, okay, once the cells are ready, we have to transfer them onto some media. So normally what is done is, we use an antibiotic free solution. Okay, after transformation we transfer them into some antibiotic free solution. Now why this is done? Okay, because during this phase, okay, the cells are going to express the antibiotic resistance gene which has been introduced because of the newly introduced plasmid. Okay, and this is going to enhance the cloning efficiency. Okay, so this is the reason it is done. Okay, now especially in case of your so especially in case of your electroporated cells, this has to be done very quickly. Okay, growing them in some nutrient media is should be very quick, because the buffers that we use for electroporation, their function is not to hold a cell or not to facilitate the cell survival for long term. So for this reason, we have to immediately carry out the growing procedure for the electroporated cells. Now these cells, okay, competent cells can be frozen also and kept for some time. Okay, so we can store them under liquid nitrogen and use them whenever we want. But again, there are chances that this whole process is going to decrease the cloning efficiency. So we have to think of that also. Okay. So ideally, it's always better that we use the freshly prepared competent cells for our whatever process we are interested in. So next is going to be your electroporation. So in electroporation also the purpose is same. Okay, if you see this picture, alright. So we have these cells placed onto the ice and we are introducing the plasmids into it, plasmid DNA, and this is transferred into a special cuvette, okay, which will be inserted in this electroporator. Now if you see this picture here, okay, the plasmids are still outside the cell. Okay, this is because of the nature of the plasma membrane, the charges on both the plasmid as well as the cell. Now this is introduced into the electroporator where we give, okay, the electric pulses for some predetermined time. Okay. So now this is decided based on the literature, the nature of the cell, the nature of the DNA, all these things will be put into picture, they will be taken into consideration, and then only we will decide the duration or the conditions to be followed during this electroporation. Now if you see here, there is some dotted line, okay, indicating the formation of transient pores into the bacterial cell, okay, the bacterial plasma membrane, so which will allow the plasmids to enter inside the cell, and okay, after that it is recovered by again placing them onto the glass, which will help to close the transient pores which are formed during the electroporation step. So this is how we will get the transformed cells, okay, or the competent cells. Now the cell is transformed. We have made the cell competent till here, and because of which the plasmid has entered inside the bacterial cell, and now we have a transformed cell ready with us. So now we have seen how does transformation occur, right? So now next we shall see the method for selecting the recombinant transformants. Okay. So what is recombinant transformants? Recombinant transformants will be those bacteria which have undergone transformation along with the newly introduced gene. So both the things are present over here, that is the newly introduced gene as well as the plasmid has entered inside the bacterial cell. So such a condition is called as your recombinant transformants. Yes. So with this I will stop over here, and next session we will see the selection procedure. Okay, so I thought of making very short videos so that you can concentrate more on one small, short duration, and you can try to understand and then move on to the next part, because next thing will be a little bit more in depth, so I don't want you to get confused. So till then, keep studying and keep smiling. Thank you.